The title of this talk is Physiology of the Vestibular System. These are the class objectives. You do not need to read them. We will use them to chart our talk. The first question is, the bony labyrinth is made of compacted petrous bone. A true, B false. This figure shows the position of the labyrinth in relation to the ear. This is a more schematic representation of the same region. In it, I want to point out to you the ganglion or ganglia of Scarpa, where the bipolar vestibular neurons live. These neurons' distal processes make contact with the vestibular sensory organ, forming the vestibular root or roots, and their proximal processes form the vestibular nerve. This is a photograph of Dr. Scarpa, for whom the ganglion for the bipolar neurons of the vestibular system were named. After his death, his assistant, Carlo Beochin, performed an autopsy and, as homage to Dr. Scarpa, removed Scarpa's head and exhibited at the Pavia University Historical Museum. It was still there in 1992. This is a drawing of the compacted bone that forms the bony labyrinth. In it, we can see the concha, where the organ of Corti lives, the vestibule, and the semicircular canals, the superior or anterior, lateral or horizontal, and the posterior one. I am now representing a lateral view of the head and a similar view of the vestibule and semicircular canals. Now, I'd like to call your attention to the lateral or horizontal semicircular canal here. Notice its position in reference to the axial plane. The axis of the lateral canal, indicated by the dashed line, is in a 30-degree angle to the axial plane, indicated by the solid line. This position corresponds to the position most of us hold a pencil on top of our ear. This is a view of the head from above. I will use it to indicate the position of the superior and posterior semicircular canals. Notice that the right superior semicircular canal and the left posterior semicircular canal axes are parallel to each other and in a 45-degree angle to the sagittal plane. Hence, they work as a pair. The right posterior semicircular canal and the left superior semicircular canal also work as a pair. They are aligned in a 45-degree angle to the sagittal plane. Other structures that can be identified in these drawings are the facial nerve canal, the oval window for the foot plate of the stapedius, and the round window. This is a simplified version of the bony labyrinth, which I like to use to stress that the labyrinth is a hollow organ. Here I have introduced a view of the lower half of the skull. I have now indicated the area of interest, which I have enlarged in this frame. The arrow points to the internal auditory meatus. The eighth nerve leaves the skull through there to contact the cochlear and vestibular apparatus. So the answer to this question is true. Next question. The endolymph has the same composition as the CSF. A true, B false. So we are going back to the bony labyrinth that, as we said, is a hollow space. Its cavity is filled with perilymph. The composition of the perilymph is similar to the cerebrospinal fluid. Sodium is about 140, potassium 4 to 5, calcium 1.2, and has a designated electrical potential of 0. This figure represents a cast of the perilymph. Here I have introduced the middle ear, the tympanic membrane, the malleus, the incus, the stapedius, the membrane that constitute the seal of the oval window, and the membrane that constitute the seal of the round window. I am now indicating to the Eustachian tube. I have now added the petrous bone, the cochlear aqueduct, and the vestibular aqueduct. The gray line I have just added represents the dura. Notice that the cochlear aqueduct goes through it, but not the vestibular aqueduct. We can now see the brain and the pia. Between the pia and the dura, we have the supranuclear space, which is full of CSF. Notice the communication of the perilymphatic sac through the cochlear aqueduct to the CSF. Now, I want to call your attention to the concha. The structure I just added is called the cochlear duct. I hope you recognize it from a prior lecture. The cochlear duct contains endolymph and houses the basilar membrane over which the organ of Corti rests. Now I want to call your attention to the other side. I have just added the section of the membranous labyrinth that corresponds to the vestibular and semicircular canal components of the vestibular system. The membranous labyrinth is made of an outer wall composed of mesenchymal connective tissue, and in its inner surface we find ectodermal epithelium. This structure contains endolymph. The composition of the endolymph resembles but is not the same as intracellular fluid. Sodium is low from 1 to 5, potassium is high about 150, calcium is low, and the electrical potential is plus 80 when compared with the perilymph. The endolymph is produced by the stria vascularis, a rate of capillaries in the cochlear duct. The cochlear duct is, as you know, in the concha. So the answer to this question is false. Next question. How many neuroepithelial organs are in each vestibular system? A3, B5, C7, D1. The vestibular neuroepithelial organ consists of supportive cells and hair cells. These organs are found at a specific location in the vestibular apparatus. This figure shows the perilymph bathing the membranous labyrinth full of endolymph. The structure here indicated is called the utricle, which means little bag. The structure here indicated is called the saccule, which means little sac. There are 3 semicircular canals, and each canal contains a semicircular duct and an ampulla. One is called the lateral duct because it is inside the lateral canal. Anterior, we have the ampulla. The second one is called the posterior semicircular duct and the ampulla. The third one is called the superior duct and ampulla. Another important structure in this drawing is the endolymphatic sac, where endolymph is absorbed. I hope by now you can recognize all these structures. I will now proceed to tell you where the neuroepithelial organs are. Unlike a single neuroepithelial organ for vision, the retina, or a single neuroepithelial organ for hearing, the organ of Corti, balanced neuroepithelial organs are scattered in multiple places. The neuroepithelial organs in the utricle and saccule are called otolith receptors or macula. The structure I have just added is the saccular neuroepithelium or macula. The macula of the saccule has a vertical orientation, thus it is perpendicular to the base of the skull. The macula of the saccule mainly responds to down or up acceleration or deceleration, as to when we are riding in an elevator. I am going back to the general figure to add the second otolith receptor, this time in the utricle. This is the macula of the utricle. The macula of the utricle is horizontally placed, hence parallel to the base of the skull. It responds to head tilt and linear acceleration. The other neuroepithelial organs are in the ampulla, as indicated here. There are 3. They are called crista ampullaris. The crista ampullaris organs do not respond to gravity. They respond to rotation. So the answer to this question is B. The connections of the vestibular nucleus include dash A, the nucleus of the solitarius tract, B, the cerebellum, C, spinal cord anterior horn neurons, D, all of the above. The vestibular nucleus complex, usually referred to as the vestibular nucleus or vestibular nuclei, can be studied based on anatomy, function, and connectivity. Anatomically, the vestibular nucleus complex can be divided into 4 nucleus or subnucleus: the superior, inferior, medial, and lateral. They are located under the 4th ventricle in the rostral medulla and caudal pons, as depicted in this frame. Functionally, we can talk about the vestibular nucleus being divided in 3 areas: an upper area involved with extraocular movements, a middle area involved with balance and posture, and an inferior area involved with autonomic control. Connectivity is by far more complex than anatomy and function subdivisions. This figure summarizes the connectivity of the vestibular nucleus. I will address each of them separately and hopefully make some sense of it. The vestibular apparatus, the labyrinth, is connected with the vestibular nucleus in a very specific way. The origin of this connection is the efferent axon of the neurons in the vestibular ganglia, through which electricity from the hair cells travel to the vestibular nucleus. This frame depicts a medullary cut at the level of the vestibular nuclei. I have now added the vestibular apparatus. The new structure I have added are the superior part of the vestibular nerve, the inferior part of the vestibular nerve, the ganglion or ganglia of Sparta, the vestibular nerve, the auditory nerve, and the vestibulocochlear nerve, and the vestibular nuclear complex. The connections between the vestibular apparatus and the vestibular nucleus are, as previously mentioned, are not random, as you will soon see. I will use the figure in this frame to explain them. In it, we can see the vestibular nucleus complex. The vestibular apparatus is shaded in different colors: green for the saccule, blue for the utricle, and magenta for the semicircular canal. As you can see, each semicircular canal has a number. The third element in this frame is the representation of neuroepithelium present in the macula of the saccule and utricle and in the crista ampullaris of each semicircular duct. Notice the hair cell, of which there are two types. Each one has two types of connections. Here in purple, you can see the input connection, and in blue, the output or afferent connections. Most of these afferent connections go to the vestibular nucleus in the way I will describe in the next few frames. The medial group receive fibers from the crista ampullaris of the lateral semicircular duct and also from the macula of the utricle go to the medial nucleus. Afferents to the superior vestibular nucleus come from the crista ampullaris of the superior and posterior ducts. Afferents for the lateral nucleus come from the crista ampullaris of all three ampullas, from the macula of the utricle, and from the macula of the saccule. Afferents to the inferior vestibular nucleus arise from the crista ampullaris of possibly all three ampulls, from the macula of the utricle, and from the saccule. This nucleus also sends fibers to all other vestibular nucleus in the same complex. In addition to the vestibular apparatus sending fibers to the vestibular nuclear complex, the vestibular apparatus sends fibers directly to the cerebellum. The cerebellum also sends fibers to the vestibular nuclear complex. In this frame, you can see the vestibular nuclear complex and the cerebellum. The red areas in the cerebellum constitute the vestibular cerebellum. The arrow points to the anterior vermis, the cortex of the vermis, and the flocculonodular lobe. I have now added the fastigial nucleus and the nuclei interpositus in front of the cerebellum instead of in the cerebellum to better view the cerebellar vestibular connections. Probably most of the fibers from the cerebellum end in the lateral vestibular nucleus. Fibers from the cerebellar vermis and the flocculonodular lobe contact the fastigial and the nuclei interpositus, and from these nuclei fibers cross to the opposite side and reach the vestibular nuclear complex through the justa restiform body, while other fibers go directly from the cerebellar cortex and the nodulofloccular lobe to the vestibular nucleus. The anterior vermis appears to be involved with vestibular spinal reflexes. The flocculi deals with the gain of vestibular ocular reflex, and the nodulus with the duration of the vestibular ocular reflex. It stems from the later comments that the cerebellum brings proprioception and vision-related information to the vestibular nuclear complex. I like to bring to your attention that this expanded interpretation of the vestibular cerebellum differs from the one we have previously described. I have included it here because some textbooks, when talking about the vestibular cerebellum, include the areas previously mentioned based on functionality. But also remember that the flocculonodular lobe of the cerebellum has two unique characteristics. The first one is that it receives input from the vestibular nerve, in addition to receiving fibers from the vestibular nuclei. And the second is that some of the Purkinje cells axons from the flocculonodular lobe leave the cerebellum bypassing the cerebellar nuclei to synapse in portions of the vestibular nuclear complex. The opposite vestibular complex sends afferent to its counterpart on the other side, as indicated here. Therefore, in addition to afferent connections, there is also efferent connections from each vestibular nucleus complex, as illustrated in this frame. Efferent connections also go to the brainstem nuclei. As you recall, when we spoke about the functional division of the vestibular nuclear complex, we mentioned that part of the vestibular nuclear complex deals with autonomic control. Hence, this part of the vestibular nuclear complex connects with the nucleus of the tractus solitarius and the dorsal motor nucleus of the vagus. I have added a transverse cut of the medulla at the indicated level and placed a rectangle and enlarged the area of interest to indicate the position of the nucleus of the tractus solitarius. Remember that this is the only cranial nerve nucleus with the nucleus surrounding the tract, and also the dorsal motor nucleus of the vagus. Another important connection appears to be with the area postrema, since vestibular pathology is frequently associated with vomiting. Also in the brainstem, vestibular efferent make contact with nuclei for extraocular muscles. There are many different models to explain these connections. I will put forth a version which I clinically find sound. Vertical eye movement pathways and lateral eye movement pathways are at least in part different. The lateral movement pathway is clinically more useful. Hence, in this talk, I will only talk about this pathway. This frame presents a clinically useful version of the extraocular muscle vestibular connections for lateral eye movements. The vestibular nuclei are simultaneously working all the time. The vertical lines indicate the frequency of the discharges. The crista ampularis of the horizontal or lateral semicircular duct appears to play a prominent role in the control of lateral ocular movements. The cranial nerve 8 connects the vestibular apparatus with the vestibular nucleus, as we know. From the vestibular nucleus, by way of the ascending tract of daters, the electrical activity connects with the contralateral parapointal reticular formation. Fibers from the parapointal reticular formation, probably climbing as part of the medial longitudinal fascicle, contact the nucleus of the ipsilateral 6th cranial nerve. Some authors omit this connection by placing parapointal reticular formation-like neurons within the 6th cranial nerve nucleus. Be it as it may, from this nucleus, fibers through the abducens nerve supplies the lateral rectus muscle. Also, from the nucleus of the 6th cranial nerve, or perhaps from the parapointal reticular formation, the later possibility not shown here, fibers cross the midline, join the medial longitudinal fascicle to innervate the contralateral 3rd nerve nucleus. The 3rd nerve nucleus gives rise to the 3rd cranial nerve. This nerve innervates the medial rectus. So now that we have talked about the vestibular nucleus connection with the extraocular muscle nuclei, we will address the spinal connections. Axons from neurons in the medial vestibular nucleus give rise to the medial vestibular tract that travels within and forms part of the caudal extension of the medial longitudinal fascicle. These tracts are concerned with head and neck coordination, especially with body-eye coordination. Axons from the lateral vestibular nucleus form the lateral vestibular tract. This tract coordinates reflexive trunk tone and proximal limb extensor tone. After spinal connections, we will address vestibular nuclear connections with the cerebellum. Vestibular cerebellar connections appear to arise from all vestibular nucleus except from the lateral nucleus. They reach the cerebellum by way of the juxtarespirum body. As with efferent connections are regional. Those to the anterior vermis are concerned with vestibular spinal reflexes. Those with floculi are concerned with gain of vestibular ocular reflex, and those with the nodule are concerned with vestibulo-ocular reflex duration. Having finished with the cerebral connections, we will address vestibular apparatus connections. The vestibular nucleus sends fibers to the neuroepithelium of the saccular and utricle macules and to the crista ampularis in the semicircular ducts. These fibers greatly influence the signal arising from the hair cells. So the answer to this question is D. Next question. Which of the following structures is less likely to be involved in vestibular information? A. Hippocampus. B. Thalamus. C. Parietotemporal junction. D. Temporal pole. There is a lot of uncertainty as to the cerebral connections of the vestibular apparatus because of three facts. Number one, there is no dissectable pathway from the vestibular nucleus to the thalamus. Number two, there is no consistently designed thalamic nucleus for the vestibular apparatus, as with the lateral geniculate for vision or the medial geniculate for hearing. Although some authors have pointed to the ventral posterior nucleus of the thalamus, probably the ventral posterior medial nucleus of the thalamus, as housing vestibular neurons. Fact 3. There is no single isolated cortical area of the brain that deals with vestibular function or balance. Different cortical areas have been named by different authors, but all agree that vestibular cortical areas are scattered in the cerebral cortex. Most consistent areas named are the parietoinsular vestibular cortex, the parietotemporal junction, area 2V, and area 3A. The hippocampus has also been cited as housing vestibular neurons. So the answer to this question is D. Next question. Otoconia are present in the crista ampularis. A true, B false. This is a segment of the Peter's bone. The redder area is the bony labyrinth. This is the membranous labyrinth, of which this is a cartoonish representation. The membrane labyrinth consists of mesenchymal connective tissue layer and another layer of cuboidal ectodermal epithelium. This is the space between the bony labyrinth and the membranous labyrinth. In it, as you know, we find the perilymph. The space inside the membranous labyrinth is occupied by the endolymph. The arrow points now to the cuboidal ectodermal epithelium. Notice how gradually the cuboidal epithelium becomes taller, forming a columnar epithelium. Also notice that slightly separated from the columnar epithelium, you see a thin layer. This layer is formed by otoconia. The otoconia are little stones made of calcium carbonate crystals. In the cartoon I just added, you can see the elements and transition I have just mentioned. The cuboidal ectodermal epithelium becomes columnar. The gelatinous space representing the space between the columnar epithelium and the otoconia, and on top of the gelatinous material you see the otoconia forming the otolith layer. I have here enlarged the cartoon so you can better appreciate the otoconia, the gelatinous matrix, as well as hair cells embedded in the columnar epithelium and nerve endings here. A further look at the pathological specimen I have shown you before will let us see the crista ampularis coccygitally. This cartoonish representation gives you a better idea why it is called a crest. Here you can see represented a segment of the crest, the cupula. Here you can see hair cells as well as columnar epithelium, often referred to as supporting cells. You can also see the nerve endings. I have placed a segment of the macula in a segment of the crypta ampularis to highlight the difference and similarities. The similarities are ectodermal cells, nerve endings, supporting cells, hair cells, and gelatinous matrix. The difference has to do with their different shapes, location, and orientation. But probably the most important difference is the presence of otoconia or otolith in the macula. Otoconia makes the macula responsive to gravity. Macular organs are often referred to as static labyrinths since they are involved with gravity. The crista ampularis are often referred to as the kinetic labyrinth. The macula is also involved in linear acceleration. The crista ampularis is involved with angular acceleration. So the answer to this question is B. Next question. Tilting of the stereocilia towards the kinocilium produces depolarization of the hair cell. A true, B false. In the process of answering this question, I would like to go through a set of facts. Number 1, hair cells are bathed in endolymph and perilymph. This is the histological specimen I showed you before. Endolymph bathes the surface of the hair cell in the macula and the crista ampularis. The composition of the endolymph is similar but not the same as the intracellular fluid. Potassium is high and sodium is low. On the other hand, the depth of the hair cells in the crista ampularis and the macula are bathed by perilymph. Perilymph is much like CSF. Sodium is high, potassium is low. Second fact, there are two types of hair cells in the vestibular neuroepithelium. This is a representation of two types of hair cells, the surface being bathed by endolymph, the rest by perilymph. Type 1 cells tend to be rounder, and the afferent nerve, here represented in blue, forms a calyx around it. Here I am indicating to the kinocilia and now to the stereocilia. The black lines represent the area of neurotransmitter release by the hair cell. This is the afferent connection taking the signal centrally. This represents the efferent connection bringing signal to the hair cell. On the other side we have the type 2 fibers. As you can see, look from this perspective, they are quite different, but they have the same components we mentioned for type 1 hair cells. How are they functionally different is a mystery. Yet, look from above, all hair cells look about the same. We find one kinocilium, a non-motile structure, and multiple stereocilia, which are motile structures. A cut, as indicated by the aqua line, will yield this view. Although this is a representation of type 2 fibers, since they both work just about the same way, I will use it to explain the mechanical workings not only of type 2, but also of type 1 hair cells. The third fact we need to know is that hair cells have a spontaneous depolarization. I hope you recall when I show you this figure that I mentioned that hair cells are always discharging at a low rate. Here you can see a hair cell with neurotransmitter, likely glutamate and aspartate, close to the synaptic sites. Here I am depicting the release of neurotransmitter that occurs spontaneously. The fourth fact is that hair cells undergo induced depolarization when the stereocilia move towards the kinocilia. So this is a hair cell. I will now enlarge this area. As you can see here, this is the kinocilium, and the shorter ones without a black line are the stereocilia. But in addition, at the tip of the stereocilia, we find strands of fibers that link the stereocilia. These strands are called tip links. In this frame, I have placed a magenta circle to call your attention to the area inside it, which I have now enlarged. In it, as you can see, we have a transduction potassium channel. Here, the channel is closed. When the stereocilia move towards the kinocilium, the displacement of the hair cell at the base exposes the transduction potassium channels to the endolymph, and potassium from the potassium-rich endolymph move into the cell, propelled by more negativity intracellular space in relation to the endolymph and by a higher concentration of potassium in the endolymph than in the intracellular space. Hence, potassium goes into the cell. Increasing the amount of potassium, that is of positive charges, in the intracellular space results in depolarization. Fifth fact, hair cell depolarization leads to neurotransmitter release. Now let's go back to the hair cell, but instead of looking at the hairy surface, look at the opposite pole, which I have enlarged in this frame. When the cell is in a polarized state, all the neurotransmitters are in. I would like now to call your attention to the area inside the circle, which I have now enlarged and added a voltage-gated calcium channel with its gates closed. The gate is represented by the yellow bar. The little circle on the side of the channel has a negative sign to indicate that it is a voltage-gated channel and that the current intracellular medium is negative to the channel opening threshold. The perilymph is a calcium-rich environment in relation to the inside of the cell. Thus, when the polarization reaches the threshold of the voltage-gated calcium channel, these receptors open, as illustrated here, and calcium goes inside. The hair cell then releases neurotransmitters. Sixth fact, hair cells hyperpolarize as the result of stereocilia bending away from the kinocilia. Movement of the stereocilia towards the kinocilium produces depolarization. Movement of the stereocilia away from the kinocilia produces hyperpolarization, thus stopping induced depolarization and decreasing the frequency of spontaneous depolarization of the hair cell. Seventh fact, hair cells, like cars, have gears. When the stereocilia are tilted towards the kinocilium all the way, the hair cells discharge very fast. When the stereocilia are less tilted, the hair cells discharge less fast. When the stereocilia are parallel to the kinocilium, the hair cells discharge even less frequently, and when the stereocilia is tilted away from the kinocilium, even less. So the answer to this question is true. Next question. In the saccule, the kinocilia are oriented away from the striola. A true, B false. Previously, we said that the macula of the saccule is perpendicular to the base of the skull, and therefore, because of its vertical arrangement, it responds to up and down acceleration and decelerations. But the situation is more complex than that, because in addition to straight up and down movements, we have up and down angular movements. To detect these changes, the macula of the saccule relies on the organization of the hair cells. This organization revolves around a structure called the striola. The striola is a curvilinear structure that runs in the midline of the macula, as indicated in this frame. The striola of the macula of the saccule forms a groove in the surface of the macula. We will now see the relation between the striola and the hair cells. In this frame, I have brought back the look of the hair cell from the top. In this new frame, the kinocilium is represented in red and the stereocilia in yellow. I have now isolated a row that includes the kinocilium to better depict the orientation of the kinocilium and the stereocilia in the macula of the saccule. As you can see, the kinocilia is away from the striola. This arrangement somehow renders the saccular sensitive to straight and angular up and down body displacement. So the answer to this question is true. Next question. In the utricle, the kinocilia are oriented towards the striola. A true, B false. This figure represents the macula of the utricle. The striola in the utricle forms a ridge and not a groove as in the macula. The kinocilia are oriented towards the striola. Again, this arrangement renders the macula of the utricle sensitive to linear acceleration and deceleration in all angles. Take a few seconds to look at this frame. The mnemonic master is giving you a clue as to how to remember the relation between the kinocilia and the striola. Notice that the second letter of saccule is A for away, and the second letter of utricle is T for toward. So the answer to this question is true. Next question. Lifting the head 30 degrees places the lateral canal perpendicular to the ground. A true, B false. This represents the head in a laying down position. The labyrinth in this position has the orientation given in this frame. The horizontal semicircular canal is closest to the tympanic membrane, and its anterior pole, where the ampulla is rostral. Also notice the axis of the horizontal semicircular canal. Here I have isolated the horizontal semicircular canal to highlight that in this position it is not perpendicular to the ground, but were we to raise the end of the bed 30 degrees, then the horizontal semicircular canal would be perpendicular to the ground. So the answer to this question is true. Next question. Ear canal irrigation with warm water increases the vestibular apparatus discharges. A true, B false. I will first give you the short version. When the head is lifted 30 degrees from reclining position, the lateral canal is perpendicular to the ground and the crista ampullaris at the superior end of the semicircular duct. The discharge frequency of hair cells of the crista ampullaris of the right lateral canal is relatively low, and so is the one from the left, since, as you recall, the crista ampullaris is not sensitive to gravity. Warm water in the right ear increases the firing rate of the hair cells in the crista ampullaris of the right horizontal semicircular duct. That was the short version. Now I will go with the long version that explains why this is so. I will start this long version by telling you what happens in a comatose patient with an intact brainstem. The figure I have just added represents the right vestibular apparatus. The crista ampullaris of the lateral semicircular canal is detailed. The structure I have added represents the external ear canal, and the arrow points to the tympanic membrane. The element I have added is meant to indicate that the external ear canal is more anterior than the most anterior part of the lateral semicircular canal, where the ampulla is, and that the crypta ampullaris of the lateral semicircular duct is more anterior than the other end of the semicircular canal, and certainly more anterior than the most posterior segment of the horizontal semicircular canal. I have now added the eyeballs looking straight ahead. When hot water is placed in the external ear canal, the fluids in the semicircular canal develop a temperature gradient. In the back of the duct, the endolymph is colder, hence heavier. The endolymph in the anterior part of the semicircular duct is hotter, hence less dense than the cold endolymph in the back of the semicircular duct. Thus, the anterior endolymph is lighter than the endolymph in the back part of the lateral semicircular canal. Hence, heavy cold endolymph drops pulled by gravity, as represented by the white arrow, whereas raises, as indicated by the red arrow, producing an endolymph flow in the direction indicated by the arrows, and tilting the crista towards the utricle. Displacement of the crista towards the utricle produces excitations, since in the crista ampullaris of the horizontal semicircular duct, the kinocilia are proximal to the utricle, and as you know, when the stereocilia tilts toward the kinocilia, the hair cells depolarize and increase their firing rate. I have now added the brainstem vestibular ocular system concerned with lateral eye movements to indicate the increased discharge rate produced by warm water irrigation of the external ear canal. So the answer to this question is true. Next question. In a comatose patient with normal brainstem, you would expect for eyes to move towards the ear being irrigated with warm water. A true, B false. Irrigating an ear with warm water increases the discharge rate of the vestibular apparatus. The discharge rate of the vestibular apparatus on the side of the ear being irrigated. Hence, in this case, the right vestibular apparatus. This will lead to excitation of the right vestibular nucleus, which will be transmitted through the ascending tract of the ethers to the left PPRF, which will excite the ipsilateral 6th cranial nerve nucleus, which will send a signal through the abducens nerve and produce contraction of the left lateral rectus. Activation of the left 6th cranial nerve nucleus will also send a signal in the medial lemniscus fascicle that will cross the midline and activate the right cranial nerve 3rd nucleus, which will send a signal through the oculomotor nerve and provoke the contraction of the right medial rectus. This constellation of events will produce slow eye movements in the direction opposite to the ear canal that was irrigated with warm water, as you can see in this frame. So the answer to this question is false. Next question. In a comatose patient with normal brainstem, you would expect for the eyes to move towards the ear being irrigated with cold water. A true, B false. Here is the same figure I initially used to answer the previous question. Both vestibular apparatus are discharging at the same rate. Cold water irrigation will create a temperature gradient in the lateral semicircular duct. The region of the ampulla, which is anterior, will be colder than the region at the other end of the semicircular canal. As the result, a flow of endolymph will be created, which will displace the cupula away from the utriculum, that is, the stereocilia tilt away from the kinocilia. This will result in a decreased discharging rate of the vestibular apparatus on the irrigated ear. This will be translated as an increased excitation of the contralateral vestibular nucleus, in this case the left, which will, through the ascending tract of the ethers, stimulate the right parapontal reticular formation, then the right sixth cranial nerve nucleus on the same side as the parapontal reticular formation will be excited, then through the right abducens, the right lateral rectus will be activated. In addition, the right cranial nerve 6 nucleus will, through the medial longitudinal fascicle, activate the left medial rectus nucleus in the third cranial nerve nucleus, and from this nucleus the third cranial nerve will carry electrical activity that will activate and contract the medial rectus of the left eye. This constellation of events will produce slow eye movements in the direction of the irrigated ear as it is being displayed in this animation. So the answer to this question is true. Next question. A patient with a normal vestibular ocular caloric testing will develop nystagmus. A true, B false. In the previous discussion, we have talked about caloric testing in comatose patients with normal brainstem function. Now we will address what happens in a normal awake patient if we were to do caloric testing. The cochlear vestibular apparatus, is now represented in the head in accordance with its anatomical position. Closest to the ear is the lateral or horizontal semicircular canal, the crista ampullaris in relation to the posterior end of the canal, and certainly in relation to the nadir or lower point of the canal. I have added a facial view of the patient in this frame and an otoscope. The otoscope is to look and make sure that the tympanic membrane is intact. Then a rubber needle connected to a syringe with very cold water, which we do not use in normal awake patients because it's distressing, but if used to irrigate the right ear canal as depicted here. The result will be that endolymph will flow towards the inferior part of the semicircular duct, tilting the cupula of the crista away from the utricle, thus inhibiting the right vestibular nucleus discharge. After such procedures, the eyes will slowly move towards the right ear, the one being irrigated, as you can see in this animation if you keep on looking at the eyes. Once irrigation stops and the eye have lateral eyes, we will see a quick return of the eyes to the primary position, that is straight ahead. This to and from movement of the eye will continue for a while after irrigation stops. As you will see in the next few frames, the eyes slowly moving towards the irrigated ear as seen here, and a quick return to the eye primary position. The line diagram I have just added depicts this process. The slow phase is traced as the eyes slowly move to the right till the eye reach maximal lateralization. Then a quick drop indicating the quick return of the eye to the primary position. Since the return phase is the one used to label nystagmus, these movements are called left-beaten nystagmus. The mnemonic for caloric-induced eye movements is CAUS, cold other, warm same. When cold water is used, the fast phase of nystagmus is to the other side of the ear being irrigated. When warm water is used, the fast component is towards the same side. Warm water caloric testing is most often used in non-comatose patients because cold water testing is distressing. But the question is, what triggers the fast component? This is an anterior view of the brain. In this area, we find the frontal saccadic eye movement area. These are cranial nerve 6, and in the parenchyma of the pons, we find the parapointal reticular formation. This is an schematic representation of the anterior brain. I have introduced the PPRF, now the right frontal area for saccadic eye movements, or area 8, which communicates with the left PPRF. Now I have introduced the left cortical area 8, which communicates with the right PPRF. I will now bring the figure that represents the response to called calorics in the right ear. Notice the position of the crista ampularis and the eye deviation towards the ear being irrigated with cold water. I have now incorporated the right area 8. As you see, the right area 8 is connected with the left parapointed reticular formation, and the left area 8 is connected to the right parapointed reticular formation. The cerebral connections have the ability to overwrite the vestibular input to the parapointed reticular formation. Thus, it brings about an involuntary fast eye jerk that returns the eye to the primary position, that is, to looking straight ahead. So the answer to this question is true. Thank you very much for your attention.